These are exactly the kind of questions you're going to get on exam 1. At what point do sodium channels begin to open? What? Oh, we're not getting that specific in this class. That's 412 physiology 412G specific. Voltage sensitive, we'll just leave it at that. 130 out of 131, somebody guess something? Okay. Okay. At threshold. At threshold. On this graph, minus 55 is that magical threshold when voltage sensitive sodium channels open up. Sodium rushes in to the neuron. The inside becomes much more positively charged than the outside. And that's what this big peak up to plus 40 shows. All right, was that 5? 2, 3, 4. Okay, last one. And always remember too, in the exam, you go at your own pace. Questions aren't popped up for just 60 seconds. You go at your own pace. There's no timing on the exam. "20 seconds. It's totally okay if you talk to each other. Or could actually answer the question together. Not in the exam, but today. Okay. Good. 100, hyperpolarizing. Everybody okay with what hyperpolarized means? Chloride flows into a cell. It's one of the means by which alcohol produces its central nervous system depressant effects. Cl minus, the inside becomes more negative with regard to compared to the outside of the cell. So it's hyperpolarized, meaning the inside and outside are even more far apart in charge. Good. Okay. Don't have a ton to do today, but you're going to see a couple of fun things for demonstration purposes. Oh, come on. Jeez. Give me a second. I know. Deep breath. There we go. Okay, this is a slide. This is a slide you really need to know. Spend a lot of time on this one, please. Okay? And I even added something to the green arrow at plus 40 where potassium channels open and potassium gets kicked out of the cell to make the interior, this intracellular environment more negative. Sodium stops coming in at that point. But really know this slide for exam 1. You're going to have 4 or 5 click-on questions that are going to be worth more than 2 points. All right. This is where we left at, okay? Everything we talked about Tuesday is happening in 1/1000th of a second. Extremely rapid. Now I want to talk about where the action potential happens and where those sodium channels and potassium channels are on neurons, okay? This class assumes that you've had a background. Yeah?
In the exam, will there be a distinction between inactivated channels and closed channels?
No. That's still, the whole inactive versus closed thing is still kind of controversial. People teach it different ways, but no. If you ask that question, just by asking that question, I know you're gonna be fine. You know there's a difference, potentially. So everybody's comfortable with neuronal anatomy, right? Okay? Just as a refresher, your prereq should have prepared you for this. The length of an axon, most axons are heavily myelinated. You remember what myelin is? It's a feet of nearby oligodendrocytes that insulate the axon. It's just like the wire that insulates an electrical cord that goes to your TV, or your hair dryer, or your toaster. It speeds electrical communication because there's insulation. So the action potential is detected, the threshold detector is right there where the axon joins the neuronal body. It's the axon hillock. Hillock in Latin just means hill, because it looks like a little hill, a little V. This is where minus 55 from today's quiz has to be detected. Because the action potential, if it reaches threshold, is going to go all the way down the axon. It's like the thermostat in your apartment. Okay. But the axon doesn't just happen at once along the length of an axon. Axons happen one myelinated segment of a neuron first, then another one, and another one, and another one as you go away from the cell body, as the axon gets longer and longer. So then it would start, sorry, I went too far. Then it would go like this. If threshold's detected here, we would go up again like this. So we call that saltatory conduction because that saltatory means jumping in Latin. One section that's myelinated has an action potential, the next one has it. That's what an action potential really looks like in 1 millisecond while it's traveling down the length of an axon. Does that help? Okay, good. That's where the sodium channels come in. Where the sodium comes in through voltage sensitive sodium channels at the nodes of NVA. At plus 40, the neuron's like, wait, no more sodium. Sodium stops coming in. You remember what goes out of a neuron to repolarize? To get that action potential down, back to rest, potassium at nodes of NVA gets kicked out actively. This is brilliant little design of biology and evolution. So again, back to running the marathon. Potassium leaving a cell is really important for bringing a cell back down to rest so that it has time to restore energy, mitochondrial, bioenergetics. Don't worry about that until next 401G test next week. So if you're low on sodium, your action potentials aren't automatically coming back to rest. Or potassium, sorry, I said, it's so easy to mix the ions up. Potassium. If you're low on sodium, you don't start an action potential. Tell me if you remember this. You probably would have been in middle school. Radio station in California had a really dumb contest in the studio. You know, the morning zoo or whatever the heck it was. And it was to see, people came in from off the street, who could drink the most water in an hour. And one of the contestants died from hyponatremia. She drank so much water, she diluted the sodium concentration in her central nervous system, and her brainstem shut down. There is a danger, it's called hyponatremia, there is a danger to that. So, anyway, okay, questions on this? Okay, what happens when the action potential gets all the way to the end of the axon? What do we call that structure at the end of the axon?
Synapse.
The presynaptic synapse, the terminal bouton, there's a lot of different words for it. That's where the neurotransmitter is stored. Action potential gets to the end. Voltage sensitive calcium channels open up. Calcium helps to push synaptic vesicles to the presynaptic membrane, and then they burst and they release neurotransmitter. This is what happened when an action potential gets to the very end of an axon. So these are voltage sensitive calcium channels. That last length of axon has an action potential. That voltage-sensitive calcium channel says it's time to open up, let calcium flow in. Calcium actually causes these microtubules, or these synaptic vesicles, to get pulled along tiny microtubules to get to the end of the axon. And they burst in and release neurotransmitter. Is that ringing a bell from, okay. Next week, we talk about what happens with this neurotransmitter after it's released on cell number 2. We'll talk about that starting Tuesday, okay? 2 things happen to neurotransmitter after it's released from an action potential, right? These go to cell number 2, they bind to a receptor, and they have an effect, whatever the neurotransmitter is. What's the second thing that happens to an action potential, or happens to neurotransmitters after it's released into the synapse here? Okay. Can be metabolized, can be broken down in the synaptic cleft. What's the third thing that can happen? Okay, reuptake, okay? Neurotransmitters don't permanently bind to a receptor on cell 2. They bind for a short period of time, then they pop off, okay? This neuron just released the blue neurotransmitter, and it's got a reuptake pump to recycle that neuron that it just released to have an effect at cell number 2. It's just evolutionary efficient. So you don't have to make neurotransmitter from scratch, just recycle what you just released. And for this class in particular this semester, there's 2 really important drugs we're going to talk about, classes of drugs. What is it? So somebody who doesn't have a squishy brain, tell me what SSRIs are and how they work, based on this slide. SSRIs. What are they first? What does SSRI stand for first? Selective serotonin reuptake inhibitor, absolutely. How do those drugs work? Escitalopram and related drugs. Yeah. They block the recycling of serotonin. You want a squishy brain? Okay. See? This is also going to be important when we talk about recreational drug use, okay? If I'm a drug that inhibits the reuptake of dopamine into neurons in my brain's reward pathway, which we'll go through several weeks, what is that drug going to do? Am I going to feel more or less reward? I'm just trying to gauge what kind of backgrounds we have. If I block reuptake, let's say the blue molecule is dopamine, right? We'll talk about this in great detail in several lectures. Increases in dopamine, we tend subjectively to find as rewarding. A lot of dopamine in your brain's reward pathway. Your sense of reward is going to go up, very high up. And that's how cocaine works. Cocaine blocks the reuptake of dopamine in the brain's reward pathway. We'll talk about the nuclei and stuff later. But that's why these simple mechanisms of how neurotransmitters get made, released, act, and then recycled, that's why it's so important. The mechanism of action of a lot of drugs just comes down to these slides. Reuptake, what kind of receptor does it stimulate? Cocaine inhibits the reuptake of dopamine in the brain's reward pathway. So you feel more rewarded. Cocaine does a million other things too, but we'll talk about cocaine later. Cool? Good? Questions? Okay, and I already asked this on today's, this is the alcohol example, so I'm not going to go over this unless you have questions. There's one receptor we're going to talk about in the next 2 or 3 weeks. When it's stimulated by a neurotransmitter, chloride goes in. Alcohol also stimulates that receptor to open, allow chloride to go in. And that's hyperpolarizing. That's why it's a central nervous system depressant. Questions? But this cell stuff is really important because it's going to help you understand caffeine, THC from marijuana products, methamphetamine. You already know how cocaine works. Because we're going to keep coming back to neurotransmitters, reuptake types of receptors, okay? Now, Bobita, would you mind helping? So I want to do this for a couple of reasons. It's a really good demonstration of how important electrical impulses are in the nervous system. So we need one volunteer. Okay. You want to be the minion? Okay, come on down. Then you'll get to do your thing. Okay. So it's really important. This is a really silly, simple way. We do this a lot in schools all over central Kentucky with 5-year-olds, 18-year-olds. It's a really good demonstration of electrical impulses controlling behavior. What kind of electrodes do you want on Emily? The big ones? Okay. So Bobby's the president of NeuroCats. If you want to know more about NeuroCats, I have a card that has the QR code to contact NeuroCats. I have a lot of cards. Ulmer. Yeah. Oh, you don't do it all. Oh, okay. Oh, okay, okay. Bobby just said I wasn't going to do her middle finger right away. Bobby is good enough. Well, this will make more sense after you see the demonstration. Bobby is going to control Emily's arm simply by using her brain to tell her muscles to contract. Okay? Your dean has been through this too. At K-Week, we got her arm flapping really good over in the ballroom. It's just a good demonstration. You know, the principle, it's a lot easier to see electrical impulses this way than it is to study it through slides like this. The dean screamed first time. Because the guy that did it, JD, the really tall guy, really big guy, yeah, he turned it up really high and the dean screamed. Okay. So Bobby is just calibrating it now, so we don't go too high on Emily. Oh. Oh, okay, yeah.
Okay, we're ready.
Okay. Does it feel weird? Bobby is muscle contractions going into a little amplifier, sending an electrical signal out to simulate Emily's right arm. Crank it up. Okay. Bobby's searching for nerves. We don't have much left after this, okay? So just be patient, please, hold tight. Oh.
Nerve found the nerve.
Yep. She's turning me red. Okay, that's good.
Okay, we got it.
That's good. Good job. Good job.
Sorry, nerve was me.
Good. That was good. No, that's okay.
Okay.
Okay, you can stay down here. We're going to do, go get your surgical gloves. All right, so now we're going to talk about, very briefly, we'll get out of here early. That was awesome. The importance of where a drug works, right? So we're going to just touch on a little bit of neuroanatomy, and we'll make it relevant to drugs today, okay? So UK, we have 5 human brains across campus. I have 3.5 in my lab, locked in a cabinet, says paper towels. Emily is going to walk around with one hemisected human brain. Are we doing 2-point discrimination? Good. You can take pictures while I talk very briefly about where a drug works being really important. Okay? You can take pictures. Oh, you don't have to use that. You can drip. It's just brain juice. Half of a human brain cut in half. Later in the semester, if we have time, I'll bring, what are we doing to Korea? Okay, where all of you have had some kind of neuroanatomy, okay? Now, if we look at the three major divisions of the brain, and neuroanatomy gets much more detailed than this, take Bio 302 if you want more. If I tell you that a drug acts in the hindbrain because the receptors for that drug are there, immediately you should know we're probably going to be messing with sleep-wake cycle, movement, essential body functions like respiration, breathing, opiate overdose. As we go from the brainstem up to the outer cortex, the behaviors and the thought process control become more and more complex. So if I give you a drug that gives you deficits in executive function after a UK football game, you make bad decisions, where do you think that drug is acting? In the brainstem or the outer cortex? The outer cortex. Okay? So you can predict, if you know most of the receptors for drug X, Y, or Z are up here in the frontal lobe, taking that drug is going to impair behaviors that correspond to that neuroanatomy. Here's another really good one. Yeah, people probably want to see it afterwards. Where do hallucinogenic drugs act in the brain? Nope. Where do hallucinogenic drugs, Jason, go in the brain? What does the somatosensory cortex do? It's a cognitive incorporation of all the incoming sensory information that you're receiving as an animal. Hallucinogenic drugs stimulate one type of receptor for serotonin in the somatosensory cortex to alter our cognitive perspective of incoming sensory information. It's just that simple. Neuroscience doesn't always have to be super complex like Biochem 401G makes it sound. It's just that simple, okay? The limbic system. What does the limbic system control? Emotion, cognitive memories. Would you guess there's a lot of serotonin producing neurons in the limbic system? That's why we have SSRIs, because they function here. Okay, in Parkinson's disease, there's a loss of dopamine producing neurons in motor areas of the brain. So we will give you a dopamine receptor stimulator that helps you regulate movement in the basal ganglia. Now, you don't need to remember the individual neuroanatomy because you've already been tested on that. But it's a really important principle. Exactly what part of the nervous system of brain a drug is working in helps determine and helps you predict the resulting subjective feelings and behaviors. Okay? I don't care that you know the neuroanatomy because you've already had that in a class somewhere. Meant to take those out. Here's an important one too, okay? One class of drugs we're going to talk about that are highly abusable. Anybody remember what happens? The dorsal horn or the back? Think dorsal fin, dolphin? What type of information comes in there? Incoming sensory information. Oops, I stepped on a Lego, barefoot in the middle of the night. Where do you think opiates function to control pain? Right here. We'll talk about what level of the spinal cord later. Opiates work in the spinal cord to inhibit pain. Their rewarding effects are the result of opiates acting in a completely different part of the brain. It's much higher up, which we'll talk about when we do opiates, your brain's reward pathway. Where do opiates function? Last thing, we'll be done. Where do opiates function when you take high doses to suppress breathing and potentially stop breathing? Based on that first slide. Forebrain, midbrain, or hindbrain? Hindbrain. That is, breathing is a vegetative function. We don't do it consciously. So those are three major effects of opiates that are based on receptors for opiates being in those three different brain regions. So the effect of a drug depends on where the receptors are. So when we talk about specific classes of drug, I'm going to tell you exactly where the receptors are. Because not all receptors are everywhere in the brain. We'll start on that after exam 1. All right, questions? If you come up and talk to me after class from now on, you get a sticker if you want. You can come take a picture of the brain.